we know that there's an enormous genetic influence on how the brain wires, how your body forms. And looking at identical twins, you can see how similar they are in so many respects, down to the level of the brain. But there are still differences between those individuals. And so the question is, what is it that happens during development that causes one individual to have their brain wired up a certain way and another individual to have their brain wired a different way? I'm Ruthazer. I'm a professor of neurology and neurosurgery here at the Montreal Neurological Institute. My lab is interested in the question of how experience during early development alters brain wiring and connectivity. We actually do this, we study this, by watching the brain wiring in real time. We can do this because we study very small aquatic animals, and these animals have very well understood brain connectivity, and it's very easy to place these animals under the microscope, having labeled a few brain cells, and watch the brain cells actually growing, developing, extending connections, forming connections with one another. We can use our ability to watch the brain wiring up under conditions where we can bias the system perhaps towards an atypical developmental pathway and see just what happens differently. In turn, what we can do with our animal models is study these fundamental mechanisms and see whether we can push them with various treatments, various manipulations. And that hopefully can translate initially through our very simple animals to larger animals and finally to the human patient population, where there's a hope of developing therapeutics and early intervention. And the two main technical approaches that we're using in the lab are electrophysiology, where we record from individual cells and watch their activity. But the other one is in vivo multiphoton microscopy, which is a nonlinear optical approach that allows you to actually do optical sectioning and create 3D models of the brain. So questions that we can ask, for example, is what causes nerve cells to grow faster or to grow slower? For me, what's really important is to come up with a kind of set of rules, the recipe book for building a brain. And I think if we can generate a reasonable sort of recipe book, then we can understand what TACC really does is it allows me and my group to hear about what's going on in patient treatment, patient care, in the epidemiology, and be able to then apply that knowledge and that understanding to our own work. It motivates us. At the same time, it can provide us with directions for our studies.